Hello, my name is Lucas Valenka. Today I will be presenting on immunotherapeutic approaches in treating Alzheimer's disease. I'm going to start with the research question to provide some context, which is, do individuals with genetic predisposition or risk factors for Alzheimer's that receive immunotherapy targeting beta amyloid have a better prognosis than unimmunized populations with the same vulnerability, as measured by reduction in beta amyloid load in cerebrocortical brain areas? Here are some basics on the disease. Alzheimer's is the most common form of dementia and also the sixth leading cause of death in the United States. It presents a significant economic, psychological, and physical burden on healthcare and caregivers. Alzheimer's is predicted to cost the U.S. $203 billion in 2013, and currently there's no cure, or really even any effective therapies for Alzheimer's. So it's definitely a big problem. Okay, I know we all have to review a couple of these presentations, so I'm going to try and keep you engaged by presenting some philosophical issues that I want you to keep in mind throughout the presentation. Let's consider the mind-body problem. It asks, how does the non-physical entity of consciousness interact with the corporeal or physical matter of the brain? Or perhaps the real question is, where in the brain does this interaction occur? So think about this. We all have consciousness, right? We all can think inside our heads, and yet we also have this physical matter we can see every time we look in the mirror. Are these two separate entities, a mind state and a separate brain state, which intimately interact and can mutually influence each other? Or is there just a physical state, or maybe just a mind state? Consider Alzheimer's disease. As the neurodegeneration occurs, destroying the connections between neurons, cognitive functioning deteriorates, and the capacity for mental events like perception and memory seem to be gone. This fits in with epiphenomenalism, which postulates a one-way causal relationship with the physical state exerting influence on the mind without any reciprocation of this influence. So what happens to the mind in the absence of brain tissue? Is it still there? How does this affect an individual's ability to interact with God? What do you think? Alzheimer's disease was first described in 1906 by the German psychiatrist Alois Alzheimer. The majority of dementias and neurodegeneration in elderly populations can be attributed to Alzheimer's disease. The characterization of dementia, the specific characterization, depends on the exact pathological features that occurs in the brain, which I will describe here in the next slide. To better understand how to treat this disease, we need to review some of the underlying pathology. Alzheimer's neuropathology is likely associated with the activation of amyloidogenic pathways. I say likely because there's still some debate as to whether this is the actual mechanism conferring disease. The amyloid cascade hypothesis is the most popular, most mainstream among neuroscientists. This hypothesis, the main premise, is that processing of amyloid precursor protein causes downstream accumulation of extracellular neurotoxic oligomeric and fibrillar beta amyloid protein conformations. So what this means is that there's this protein called amyloid precursor protein, which exists in the plasma membranes of multiple tissue types, including neurons. Processing of this protein yields beta amyloid in certain isoforms. and the different amino acid lengths determines which isoform it is. Now, the most toxic and the one that's most implicated in Alzheimer's is the 42 amino acid length. So when you see beta amyloid 42, that's what they're referring to. And this particular isoform is the most implicated because it tends to be the one that promotes the most aggregation with other beta amyloid proteins. The 40 length amino acid isoform doesn't bind as much to the to other proteins. And going into some of the pathophysiology that results from activation of these amyloidogenic pathways include neuroglial cell activation. Neuroglial cells, those are the supporting cells. They're not neurons, although they come from a common progenitor cell, but they just provide a supporting role as far as helping with metabolism, nutrition, and things like that. Other pathophysiology includes calcium dyshomeostasis, synaptic dysfunction and failure, neuritic dystrophy, loss of neuritic spine density, and neuronal cell death. Now, all of this leads to the clinical signs, which include cognitive impairment, memory loss, and generally there's going to be some neuropsychiatric disease associated with Alzheimer's as well. Now, the reason why glial cell activation is pathological is because it causes inflammation that can be potentially harmful to neurons. So what does this all really mean? And it's basically just misfolding of protein. Misfolded protein likes to accumulate, and the conformations that result usually tend to be toxic. Now, proliferation of misfolded protein accompanied by a change in solubility is termed amyloidosis. So yes, Alzheimer's is a dementia and it's a neurodegenerative process, but technically, based on pathology, it's a type of amyloidosis. Now, if you work in the inpatient hospital setting, you probably deal with a type of amyloidosis every shift. I'm going to get a little bit off topic here, but I think it'll play in. Diabetes type 2 is also technically a type of amyloidosis. A protein called amylin is co-secreted with insulin by beta cells, and there's a synergism between these endocrine peptide hormones, amylin and insulin. And amylin is also created inside the cell, just like insulin, and therefore follows a secretory pathway to exit the cell. Post-translational modifications are needed to achieve the proper folding of incomplete amylin into functional amylin, and this occurs actually while amylin is exiting through these secretory pathways. However, when there's high demand for insulin, like in the hyperglycemia that occurs in diabetes, these secretory pathways can become saturated, and this exposes unfinished or unprocessed amylin in the cytoplasm, and this tends to aggregate with other unfinished amylin, creating insoluble misfolded protein fragments. These can be toxic to the cell and can destroy the beta cells. Insulin resistance, I think, is likely due to the absence of functional amylin to complement insulin. And of course, it's also due to what we learned in nursing school, which is a downregulation of insulin receptors. But eventually, beta cells will become destroyed from all this oxidative stress, from this misfolded amylin. So, it's real important. Education for lifestyle modifications for type 2 diabetes is important earlier in the disease process, because I think you can reach that point where it's too late and you actually destroy all your beta cells. Here is a visual representation of neurons in the brain. Notice the accumulation of oligomers occurring at the synaptic site on the top right of the slide. This is where neural connections are made, allowing communication through signal transduction, making cognition and mental functioning possible. Oligomers are still in the soluble phase, and they seem to aggregate around synapses. This correlates well with the synaptic failure seen in Alzheimer's. Recently, research has suggested the most neurotoxic form of amyloid is in this oligomeric soluble state. The term oligomer refers to a molecular complex consisting of a few monomers, which are single amyloid peptides. As amyloid peptides continue to non-covalently bond together, the process called polymerization, eventually an insoluble protein complex will form. This is known as plaque. Now look on top of the slide and see how the dendrite is not connected to another neuron anymore. Is the actual neuron dead? No, but if the neuron isn't connected to other neurons, does it even matter if it's dead? What's important is the connections that are established between neurons. And in Alzheimer's disease, the connections are severed. But also, in Alzheimer's, it's harder to achieve these connections. And this is why. On the previous slide, it stated neuritic dystrophy and loss of dendritic spine density as the pathophysiology of Alzheimer's. And looking at this picture, here's that depiction. The dendrites are seen shriveled up, and this is a result of the amyloid buildup, which induces toxic effects, causing remodeling of the cellular components. Eventually, a buildup of hyperphosphorylated tau occurs, and tau protein is responsible for the cytoskeletal integrity, which maintains the structure. So when that protein also becomes misfolded, that's what causes that shriveling effect. So when amyloid protein accumulates around these dendrites, it alters its three-dimensional orientation. I mean, obviously, it's not connected anymore. But I want you to conceptualize how important the specific orientation of neurons and neurites are. A dendrite can be positioned in a way that will increase the likelihood of creating a connection with another neuron. So obviously, the more shriveled up it is, less likely that it will make a connection. But this can also you can manipulate this to be a positive or a negative thing. Obviously, in the context of Alzheimer's, there's a negative outcome. The more loss of spine density, less likely to reestablish connections. But we can also use this concept in a positive way. Think about what you learned in school about the brain and neurons. You were told that neurons cannot regenerate, as in re-entering the life cycle, producing identical daughter cells. While this is only semi-true, the real question is, are neurons capable of generating new dendrites and new connections between cells? And the answer is yeah. That mechanism is called neuroplasticity. This science has recently emerged as new technology has confirmed the brain's ability to achieve this. It's always been theoretically possible and suggested, but no one really paid attention because there wasn't any empirical support for that hypothesis. So I'll also make the point that it's not just the orientation and connection of individual synapses that make cognition possible, but it's the sum of millions of neurons grouped together in neural circuits that also need to be oriented in a specific way that really allows cognitive functioning. And this is because of the electrophysiological nature of the brain, which is analogous to the conductive systems in the heart. In the heart, if myocardial cells in the atria fire at the wrong time or in the wrong direction, this can disrupt the entire function of the chambers of the heart, causing AFib. Okay, I know I'm getting a little bit off topic, but hopefully this will help you conceptualize what's happening in the brain, which will really facilitate your learning. So the whole concept of neuroplasticity has a lot of implications on rehabilitation, especially in the setting of stroke, but in the context of Alzheimer's, this really affirms the need for early recognition of the disease and early treatment, because there's still that potential for the brain to recuperate and reestablish these connections that are severed and are impaired in Alzheimer's. We have all heard of Plato, the Greek rationalist who was the founder of Western philosophy along with Aristotle and Socrates. He had a theory called the theory of forms, which asserted that non-material abstract forms possess the highest and most fundamental kind of reality. Now, forms is referring to an essence, which does not contain any mass, but is also not just a kind of energy. Of course, Einstein said energy and mass are transposable, so this is something that is distinguished from that. So when I think of his theory and apply it to neuroscience, I wonder if these forms that he's referring to are the orientation of neurons and neural circuits, which allow cognition and, above all, are responsible for consciousness in the human brain. Neurons are a material substance, yes, but they will not confer cognition unless oriented or positioned in a certain way or form. So applying this to the mind-body problem, which asks, where does the mind and consciousness meet in the brain? I believe it's in these forms that millions of neurons take. Think about how the brain holds on to a memory. Memory occurs when neurons react to an environmental stimulus and creates a connection in the brain. Memories are unique to every individual person, so these connections in the brain must also be unique. Actually, let me restate that. The actual bioneurological mechanism that connects neurons is similar in every person, but it's the orientation and conformation of these connections between millions of neurons and neural circuits that is unique. You may disagree, but in my opinion, which is based on Plato's idea of forms, or theory of forms, this is where the soul exists. René Descartes, he was the guy, that created the scientific method and the Cartesian plane. He said the soul existed in the pineal gland back in the 1600s, and obviously he's been wrong about that. So that's my theory on the mind-body problem. Going back to Alzheimer's, the majority of the meaningful research done in this context has been translational research, which the textbook states it's research that transforms scientific findings or discoveries from basic lab, clinical, or population studies into new clinical tools, processes, and applications. The purpose is to link basic research to the point of care. So, how does this fit into characterization of research type? This might just be a semantic thing, but really that's equivocal and ambiguous. It's kind of a combination of both. When you're studying things at the cellular and molecular level, trying to interpret those findings into human applicability is difficult and takes substantial amounts of empirical confirmation. For this reason, 2 of my articles were integrated reviews, which kind of just have the most important findings and really the implications of years and years of research. So currently there's no cure or really any treatment for Alzheimer's disease. The FDA has only approved 2 categories of drugs, the cholinesterase inhibitors and NMDA receptor antagonists, which really don't effectively delay the progression of the disease. So let's do a quick review on immunology. An antigen is a substance that binds to an antibody. Proteins can act as antigens. Based on the theory of the amyloid cascade hypothesis, the amyloid could be a potential antigen that antibodies could respond to. So the hypothesis, which was determined by research question, was can antibodies with affinity for amyloid antigens be used to treat Alzheimer's? Now remember, the lymphocytes are responsible for specific adaptive immunity. It's different from that innate immunity. There's been a lot of debate as to the extent of the immune surveillance in the brain, and this is because of that really tight, selectively permeable blood-brain barrier. But when you do cerebral spinal fluid cell counts, there are immune cells that do show up, but what's the extent of that surveillance? That hasn't been determined yet. So then you have to ask the question, is an immunotherapeutic approach, is that even going to be effective at treating Alzheimer's? And I'm going to present some of the research that has been done in this area. Mouse models and in vitro studies show that amyloid immunization does clear amyloid in the brain and improves cognitive function. Only a few human clinical trials with amyloid immunization have been conducted, which did reduce the amyloid load, but had variability in regard to the improvement in cognitive function. So why is there a poor correlation between amyloid clearance and cognitive function in human trials that have tested immunotherapeutic approaches? Is that because amyloid is not the true causative agent responsible for Alzheimer's? I don't think so. I think there's a lot of error in the experimental designs. Consider the fact that histological analysis can only be done on the Alzheimer brain after the person has passed away. Also, experimental designs that really have considered the timing of initiation of therapy, a lot of that is still missing in the literature. Although there are some clinical trials that are going to be starting soon that do address those issues, which I'll talk about soon here. And then on the second bullet there, it says research suggests amyloid accumulation can precede clinical signs up to 20 years. A positron emission tomography scan with amyloid-specific Pittsburgh compound B showed amyloid deposits 15 years prior to the clinical symptoms. Again, I don't think it's because amyloid is not the causative agent, but there could be subtle changes in the brain that are occurring before somebody's aware. Perhaps their capacity to learn is inhibited, but it's not like they're going to be conscientious of that. Somebody really going to know or think that they have Alzheimer's because they're not learning as well? I mean, that could potentially play part in the diagnosis there, but I don't think we're at that point yet. Early identification is key to successful treatment, especially as new therapeutic approaches are developed, and that inference can be made based on all the empirical support for amyloid accumulation occurring up to 20 years prior to manifestation of the disease. There still are limitations in diagnostic medicine as far as distinguishing from soluble and insoluble forms of amyloid, but I believe I had read that there are clinical trials underway for scientists who had proposed new ideas on how that is possible to make that discernment between the two types. But genetic analysis is probably going to be the best way for early identification of high-risk individuals, especially if there is that family history of dementia or Alzheimer's. And the genetic links to Alzheimer's are well known and well documented. Unfortunately, I wasn't able to address that in my paper, lack of time, lack of space, but the genes, we know what genes cause early Alzheimer's, and there's a strong genetic link to even the late Alzheimer's, and I won't get into that just because of so I won't extend this any further. But certainly neuroimaging for any individual showing any signs of mild cognitive impairment, just to try to see signs of amyloid accumulation as early as possible and start treatment as soon as possible. So there are 3 types of immunization targeting beta amyloid, which are currently have been tried at least on animal studies. Passive and active, we are familiar with those. In passive immunization, we inject preformed antibodies. With active immunization, we directly inject the antigen that confers disease. But the DNA immunization, that is the new breakthrough concept that has been developed and has shown, compared with all 3 types of immunization, has shown the greatest immunogenicity, safety, amyloid clearance, and cognitive improvement. Now, the DNA immunization has not been tried on humans at all, but will be. The passive and active have, and those human clinical trials are currently in phase 2 and phase 3 for development. The first human trials using active immunization actually had to be stopped after the first 2 years because there was an inflammatory response that developed in 6% of the patients. They got meningoencephalitis, but they determined that was due to a Th1 mediated immune response, and using the DNA immunization and some of the other active immunization approaches that have been developed completely they don't induce that Th1 immune response like the first clinical trials did. So we're in the process of development. The approaches are getting a lot safer, and they're showing a lot more improvements in cognitive function, and hopefully this will be the way that will actually, if not cure, at least severely delay that progression of disease. Let's talk specifically about the DNA immunization approach. Scientists at the University of Texas Southwestern Medical Center in Dallas developed this type of genetic immunization that can specifically target a beta amyloid protein. The DNA vaccine is injected with a gene gun, which is a bioballistic particle delivery system that can inject genetic material straight into the cell. The ammunition used is plasmid DNA coated around a metallic particle, which is usually gold or tungsten. So this immunization approach uses a cascade binary Gal4 upstream activating sequence beta amyloid 42 trimer vaccine system, and in the animal studies demonstrated high immunogenicity, high antibody expression that was strongly polarized with the Th2 bias, and there was no accompanying inflammatory T cell response. So remember how the Th1 mediated response caused the meningoencephalitis in the first human trials? This vaccine only elicits the Th2 T cell response, and Th2 are the helper T cells that express the CD4 proteins, and those cells secrete cytokines stimulating B cell proliferation and antibody production, therefore avoiding that Th1 response. Also, there was a limited stimulation of the T cells that express the CD8 surface proteins, and those are the cells that mediate cytotoxic immunity, which those are the cells that respond to the major histocompatibility complex 1 proteins, which all nucleated cells express, including neurons. So having an immune complex in neural tissue of that kind, that would be dangerous. But if it's just primarily a B cell response, the B cells that confer the humoral immunity, that is a safe approach, and that's what these scientists have developed, and it is just absolutely brilliant. On the next slide, we're going to go a little bit more into how the DNA vaccine actually works. Here is a model of the DNA vaccine, which uses a 2 plasmid system, hence the term binary when describing it. The activator sequence on top of the model has a cytomegalovirus promoter region, which will elicit the transcription process upon entering the cell. Downstream from there is the encoding region for a yeast Gal4 transcription factor. When expressed, this will bind to the upstream activating sequence on the responder plasmid. And since it's a transcription factor, will drive the transcription in the responder plasmid. This is the plasmid that contains the encoding region for a beta amyloid trimer, which expresses as a protein product structurally similar to 3 non-covalently bonded beta amyloid peptides. A molecular technique to enhance processing of the beta amyloid region is including encoding regions for an adenovirus E3 leader peptide in front of the amyloid trimer sequence and an endosomal targeting peptide downstream from there. In the animal studies conducted, DNA vector-coded gold particles delivered the beta amyloid antigen encoding genes into mouse dermal cells using gene gun bombardment. Exposure to dendritic cells, which migrate rapidly into draining lymph nodes, will then function as antigen-presenting cells, initiating the immune response. In conclusion, DNA vaccination has the potential to safely delay the onset, slow the rate of progression, or even prevent Alzheimer's disease, as it has shown to do so in animal studies. This is done by inducing an immune response targeting beta amyloid protein. Preclinical studies for human clinical trials using this DNA vaccination are underway. Also, clinical studies using immunotherapy on individuals with genetic predispositions prior to clinical symptoms are also set to begin soon.